Interferon type I. Human type I interferons, IFNs, are a large subgroup of interferon proteins that help regulate the activity of the immune system. Interferons bind to interferon receptors. All type I IFNs bind to a specific cell surface receptor complex known as the IFN alpha receptor, IFNR, that consists of IFNR1 and IFNR2 chains. Type I IFNs are found in all mammals, and homologous, similar, molecules have been found in birds, reptiles, amphibians and fish species. Mammalian types. The mammalian types are designated IFN alpha, alpha, IFN beta, beta, IFN kappa, kappa, IFN delta, delta, IFN epsilon, epsilon, IFN tau, tau, IFN omega, omega, and IFN zeta, zeta, also known as limitin. IFN alpha. The IFN alpha proteins are produced mainly by plasmacytoid dendritic cells, PDCS. They are mainly involved in innate immunity against viral infection. The genes responsible for their synthesis come in 13 subtypes that are called IFNA1, IFNA2, IFNA4, IFNA5, IFNA6, IFNA7, IFNA8, IFNA10, IFNA13, IFNA14, IFNA16, IFNA17, IFNA21. These genes are found together in a cluster on chromosome 9. IFN alpha is also made synthetically as medication in hairy cell leukemia. The international non-proprietary name, INN, for the product is interferon alpha. The recombinant type is interferon alpha-1 1. The pegylated types are pegylated interferon alpha 2A and pegylated interferon alpha 2B. IFN beta. The IFN beta proteins are produced in large quantities by fibroblasts. They have antiviral activity that is involved mainly in innate immune response. Two types of IFN beta have been described, IFN beta 1, IFN B1, and IFN beta 3, IFN B3. A gene designated IFN beta 2 is actually IL-6. IFN beta 1 is used as a treatment for multiple sclerosis as it reduces the relapse rate. IFN beta 1 is not an appropriate treatment for patients with progressive, non-relapsing forms of multiple sclerosis. IFN epsilon, kappa, tau, delta and zeta. IFN epsilon, kappa, tau, and zeta appear, at this time, to come in a single isoform in humans, IFN K. Only ruminants encode IFN tau, a variant of IFN omega. So far, IFN zeta is only found in mice, while a structural homologue, IFN delta is found in a diverse array of non-primate and non-rodent placental mammals. Most but not all placental mammals encode functional IFN epsilon and IFN kappa genes. IFN omega. IFN omega, although having only one functional form described to date, IFN W1, has several pseudogenies, IFN2, IFN4, IFN5, IFN9, IFN15, IFN18, and IFN19 in humans. Many non-primate placental mammals express multiple IFN omega subtypes. IFN nu. This subtype of type I IFN was recently described as a pseudogene in human, but potentially functional in the domestic cat genome. In all other genomes of non-feline placental mammals, IFN nu is a pseudogene. In some species, the pseudogene is well preserved, while in others, it is badly mutilated or is undetectable. Moreover, in the cat genome, the IFN nu promoter is deleteriously mutated. It is likely that the IFN nu gene family was rendered useless prior to mammalian diversification. Its presence on the edge of the type I IFN locus in mammals may have shielded it from obliteration, allowing its detection. Sources and functions. IFN alpha and IFN beta are secreted by many cell types including lymphocytes, NK cells, B cells and T cells, macrophages, fibroblasts, endothelial cells, osteoblasts and others. They stimulate both macrophages and NK cells to elicit an antiviral response, involving IRF3/IRF7 antiviral pathways, and are also active against tumors. Plasmacytoid dendritic cells have been identified as being the most potent producers of type I IFNs in response to antigen, and have thus been coined natural IFN-producing cells. IFN omega is released by leukocytes at the site of viral infection or tumors. IFN alpha acts as a pyrogenic factor by altering the activity of thermosensitive neurons in the hypothalamus, thus causing fever. It does this by binding to opioid receptors and eliciting the release of prostaglandin E2, PGE2. A similar mechanism is used by IFN alpha to reduce pain. IFN alpha interacts with the mu-opioid receptor to act as an analgesic. In mice, IFN beta inhibits immune cell production of growth factors, thereby slowing tumor growth, and inhibits other cells from producing vessel-producing growth factors, thereby blocking tumor angiogenesis and hindering the tumor from connecting into the blood vessel system. In both mice and human, negative regulation of type I interferon is known to be important. Few endogenous regulators have been found to elicit this important regulatory function, such as SOX1 and aryl hydrocarbon receptor interacting protein, AIP. Non-mammalian types. Avian type I IFNs have been characterized and preliminarily assigned to subtypes, IFNI, IFN2, and IFN3, but their classification into subtypes should await a more extensive characterization of avian genomes. Functional lizard type I IFNs can be found in lizard genome databases. Turtle type I IFNs have been purified, references from 1970s needed. They resemble mammalian homologs. The existence of amphibian type I IFNs have been inferred by the discovery of the genes encoding their receptor chains. They have not yet been purified, or their genes cloned. Piscine, bony fish, type IFN has been cloned first in zebrafish, and then in many other teleost species including salmon and mandarin fish. With few exceptions, and in stark contrast to avian and especially mammalian IFNs, they are present as single genes. Multiple genes are however seen in polyploid fish genomes, possibly arising from whole genome duplication. Unlike amniote IFN genes, piscine type I IFN genes contain introns, in similar positions as do their orthologs, certain interleukins. Despite this important difference, based on their 3D structure, these piscine IFNs have been assigned as type I IFNs. While in mammalian species all type I IFNs bind to a single receptor complex, the different groups of piscine type I IFNs bind to different receptor complexes. Until now several type I IFNs, IFNA, B, C, D, E, F and H, has been identified in teleost fish with as low as only one subtype in green pufferfish and as many as 6 subtypes in salmon, with an addition of recently identified novel subtype, IFN in mandarin fish. Identified novel